Hi, my name is Roy Henson. I'm going to go over Lesson 2 PowerPoint to kind of shed some light on this subject. I'm just going to read the goals here real quick and then we'll get started and kind of figure out what's important and kind of shed some light on some of these subjects. Describe the points of this lesson are: describe several non-Mendelian inheritance patterns involving a single characteristic, describe how two genes can interact with one another to determine a single phenotype, examine and influence environmental factors on phenotype, understand how complementation can be used to determine if gene interaction is occurring. So this first one is, we're just going to go over some of the vocabulary here. First one is incomplete dominance. Incomplete dominance is talking about when you have two different phenotypes. You have, well, right here you have red flowers and you have white flowers. And what happens is when you cross the two of the same species and you get this incomplete dominance, or this mixing of the two. There's a little Punnett square in here, kind of explains what happens is you end up with 50% of the heterozygous phenotype, 25% of the homozygous dominant, and 25% of the homozygous recessive. And that's incomplete dominance, is where it splits the phenotype between the two when you end up with a heterozygous individual. The next one is overdominance. Overdominance is when you have a heterozygous with an advantage or a reproductive advantage over homozygous. The example they give is sickle cell anemia. What happens is, and what's explained in this little Punnett square down here, is that you have homozygous dominant, which is unaffected, doesn't have anything to do with anything with the trait, and you have these heterozygous individuals here, and what this sickle cell anemia does is the heterozygous ends up gives you a malaria-resistant strain, and so you don't die of malaria. Then the recessive, or the homozygous recessive, is the one in the bottom, and that's where you end up with sickle cell anemia. And so this occurred, this trait of overdominance occurred over a period of time, just from natural selection. People with malaria died, and people that didn't went on to reproduce. And so it's just talking about how it overdominated because of natural selection, and the heterozygous has that. The next one is codominance, and it's talking about A, B, and O blood types. The blood type, this is actually a sugar read. It's a set of sugars on blood, and this just occurs. And so O is actually the base sugar branch here, and so everyone has these basic sugars, and that's what makes O is. And then what happens is, as time went on, there were changes in different sugars added, and so as you can see, with A sugar was added here, and that was passed on, and then with a different antigen B, a different type of sugar was added, and so you end up with these two different sugars from A type and B type, all branched off of the main branch sugar. And so if you don't have any other antigens on it, it's O. If you have one type, it's A. If you have another type, it's B. And then what happens, A and B blood types mixed through stuff, and then you ended up with AB type, which has the A antigen and the B antigen on the same blood type. And so pretty much what it talks about is this is codominance, because you can have A type or B type, or you can have AB type, a combination of the two, which is a completely different phenotype in itself, and that's what codominance is. So two alleles for a trait contribute equally to the phenotype, and that's kind of what this is explaining, that you can have one type which is completely recessive, then you can have another phenotype, and then another phenotype, and then those mixing creates a third phenotype. That's codominance, those words together. And the next one is incomplete preference and expressivity. So I'm going to kind of hit expressivity first. Expressivity is when you have variations of a phenotype specified by genotype, and so you'll have variations of a specific phenotype. So this example is protodactyl, and so high expressivity would be like having 7 fingers, and low expressivity will be having like 5.5, but the same kind of thing. It's the same genotype, phenotype. Then penitence is an individual that does not display a phenotype associated with their genotype. So someone carrying the genes for paradoxal but doesn't have any extra fingers. Kind of what I understand from that. Next one is multiple alleles. And what it's talking about with multiple alleles is you have multiple alleles for different phenotypes and different genes. So as it talks about with these rabbits here, you have the homozygous dominant, which is full color coat, which is this guy right here, and then from there it just goes down all the way to the homozygous recessive is the albino, and then it just kind of it's just an order, it's a chain. As you can see, is C is most dominant, and then the chinchilla pattern is next most dominant, and then the Himalayan is kind of medium recessive, and the most recessive is the albino. And so if you cross a full coat with an albino, all the kids are going to turn out full coat. If you cross full coat with anything, you're going to have heterozygous, and it's going to be full coat. But and the same thing goes with chinchilla with Himalayan and albino. If you cross chinchilla with Himalayan and albino, you're going to end up with chinchillas. And Himalayans will only cross with albinos to get you Himalayans, and so on. That's kind of how that works with the multiple alleles and the order, and guess the which allele is more dominant over another one. And then there's lethal alleles, and this really is a really good example of these Manx cats. And so the Manx the Manx gene is dominant, and so if you get two Manx cats, you cross them together, they won't have any kids because all the kids die, embryonic death. It's just kind of how it works. But if you end up with the heterozygous, you end up with these Manx cats, and so you end up with tailless cats. It's kind of cool. And then if if they're recessive, it's non-Manx cat, full tail, just full tail going out there in the end. And so that's kind of how that works. So if some of these sometimes it's recessive, it's homozygous recessive is lethal, and sometimes homozygous dominant's lethal. But for this example of the Manx cats, it's the homozygous dominant is the recessive allele. And it just means that they die, and so they end up with this phenotype that's 2-to-1. And so if you ever see a phenotype of 2-to-1, you know it's a lethal allele that's showing up there. And then this is the maternal effect. I thought this was really interesting. Pretty much what it's talking about. It's saying that the mother decides the phenotype. And so as you can see here, the mother of this one is homozygous dominant, and the mother of this, and then homozygous dominant, it doesn't matter. All of her heterozygous children are going to be the same phenotype as the mom. As you can see over here, this is a homozygous recessive, and then all the heterozygous children are recessive. And so this follows as this goes down. You see that you end up with this 1 to 2-to-1 ratio, and they're all the same phenotype from the mother of here, because she carries the dominant allele, and so that mother, it's the one that happens. And then as you continue to mate those, you'll end up with a third here, or a one quarter here, ended up as the other phenotype, or the recessive phenotype. And it just also goes down to the mother decides the traits. They're just going to look at them, and here's kind of how this works with the maternal effects during the mating of the eggs. And so you end up with this diploid nurse cells helping the development of the egg, and they're the ones that help doing it. And so the nurse cells come from the mom, and the mom's proteins cause it to spin the way it's spinning. I guess it's that way. And so these genes produce past fertilization. For humans, it's mitochondria. It's going to talk about a little bit more here, and just kind of how those different alleles spill in different ways, and just kind of explains it more. So if the nurse cells come from a homozygous dominant mother, all the eggs are going to be D. And then it really comes down to whether it's heterozygous. This is where it makes a big difference, because if they're homozygous recessive, they're all going to spin in the homozygous recessive manner. But if they're heterozygous, it's really going to depend on if the mother was homozygous recessive or homozygous dominant, and that's where it comes from. And so it goes on to continue to talk about maternal inheritance, and talks about the mitochondria and chloroplast and how those are inherited from their mothers. Like for humans, we inherit all of our mitochondria DNA from our mothers, and so whatever your mom's mitochondria was, that's your mitochondria DNA too. So it just talks about that, and extragenital genes are inherited strictly from the mother. Maternal inheritance, it's kind of the way it works. The phenotype of the offspring depends on the phenotype of the mother. Just what I was saying, and it just goes to explain this more about how these chloroplasts can end up mixed, and kind of in an explanation of why some of these plants are white and some of them are green, and it all comes back to the mother. And so you'll end up with some of these cases where the mom is half and half, the mom is white, and so the mother passes hers onto her son or her offspring. So I guess the key definition word you're supposed to get out of this is heteroplastic cells, or the green patch contains cells with wild-type chloroplasts and heteroplastic cells with both mutant and wild-type chloroplasts. And so that's kind of where that comes from. And then we're going to talk about gene interactions, and we're going to talk about these chickens here and these different inheritance patterns. If the inheritance pattern single trait can be expressed in 16ths, then the interactions are occurring. So these are the 16ths. And it just talks about gene interaction, how two or more genes can control a single phenotype. And this kind of goes back to the rabbits and stuff like that. And so it talks about how you can end up with this walnut comb, rose comb, pea comb, single comb just by the mixing of the two different types of genes. So one gene affects another gene, and that's called epistatus, when alleles of one gene mask the phenotypic effect of another gene. And that's kind of what's going on here. And it goes into more detail right here. And so this phenotype trees here. So if you mix two heterozygous for both genes, you're going to end up with this nice plastering of all these different things. It's actually, I'm pretty sure that's a 9 to 6 to 1 ratio. Yeah, 9 to 6 to 1, or 9 to 3 to 1. 1, 3, 3, and then and it just goes like this. These blank spots can be the dominant allele or the recessive allele. It's going to end up with the walnut comb. The same thing with the rose comb. This can be a dominant or recessive R, or a big R, little r, and it's going to end up with the rose comb. This can be the big P or little p comb, and if we're all recessive, that's the single comb. And that's really kind of how we get our skin color. So our skin color is really interesting. There's 3 different genes that mix together to code for that melanin, and so you can get a huge diversity depending on the mix. And so you can get anywhere from albino all the way to super black. And then it kind of occurs in this bell curve that when you mix these two alleles together, or you mix these 3 genes together, you're going to end up with this bell curve. And the majority of the people are going to be in the middle here. And it's kind of how it works. And then it talks about compilation tests to kind of see if something's true breeding or not. So if 2 mutants have the same phenotype, they contain mutations in the same gene or 2 separate genes. And so they make 2 true breeding mutant strains to test each other. And so you have this white gene, or this white flower and this white flower. They're both true breeding, and then you mix the 2, and then you can end up with you can figure out and see if you end up with all wild type progeny occurs when 2 mutant strands are complement each other. And so you can end up with all wild type progeny even though you have 2 true breeding strands. You mix the 2 and you end up with purple because the expression of the genes is mixed by 2 different genes. And that's kind of how that's done. That's how you test it. And it always comes out to be a 9 to 7 ratio if you end up with 2 genes that are both true breeding and you mix them. The 9 to 7 ratio is something to look out for. And so the last thing I kind of want to touch on is this influence of environmental phenotype. And so it brings up the foxes here. And so in the winter, the protein that codes for fox's fur actually changes depending on what temperature it's around. And so if it's cold, the protein tends to go more, express more of a white fur, and versus if it's hot, the protein tends to code for darker fur. And that's actually the exact opposite of what happens with Siamese cats and pugs. If it's cold, it's darker. If it's warm, it's whiter. But that's something that people have bred for. And then there's also, it talks about phenylketonuria, a disease where phenylalanine cannot be metabolized in humans, influenced by their diet. And so that's a gene that can be expressed, or kind of can express, or can change the phenotype if you just by the diet of the food. That's actually it's because they're missing a protein on that one. And then it talks about the faucet number of fruit flies influenced by temperature. So the number of little eye sockets they have is influenced by temperature. So those are just all things that is good to know for this slide. I hope I explained enough. And have a great afternoon. Again, this is Roy Henson and Bio 375 Genetics. All right.